Morning. This is the instructions for Module 8, Day 3, Wednesday, May 27th. I did have a live session yesterday, and I did not record my Period 3 live session, so I posted the Period 2 live session to both my Period 2 class, which is right here, and I also posted the same session to my Period 3 class as well. Let's just double check, make sure that that is there. Live session 8.1, Monday, May 26th. Yes, it's actually 8.2. I keep forgetting that we didn't have school on Monday. Just double check, make sure that does play. So, just so and it's just
Let's see.
Easy for me, just really quick, just to recap week 7. I'll do it, and I won't even go through the stuff. I'll do it real. All right. So that was a great session. Also, by the way, I encourage you to get a chance to take a look at that. I did a lot of instruction through that lesson. So yesterday's work, let's just take a look. And I prefer to work out of Period 2, so we'll just do that. All right. So just a recap of yesterday. Yesterday, I had you look at the structure of DNA in the cell cycle. In the notes, there's very little on the structure of DNA. Most of the experience in the structure of DNA you will have are on the nucleotides, which is the kind of like the rungs on the ladder. Most of the notes and most of my instruction yesterday was on the cell cycle. That is actually what's most important. Fundamental component to this is the cell cycle. Although that doesn't mean that I'm diminishing the importance of the structure of DNA. It's just that there's a lot of memorization components to that, and I really want you to walk away with an understanding of how cells are born and how they die and what can happen in between, because there's a lot of application to the cell cycle in diseases like cancer. All of this can be linked to cancer, and what's really fascinating are the approaches that we take towards battling cancer are interruptions that we deliberately do to the cell cycle to stop cells from growing. So pretty cool stuff. So that was yesterday. Let's just do a quick little, go to the notes here really quick. So words that you want to make sure you're aware of: mitosis and cytokinesis. Remember, cytokinesis is the complete splitting of the cells. Mitosis is the actual division process. We talked a little bit about the chromosome, the structure that's in the middle called the centromere. Some highlights, some stuff. And then the cell cycle. This diagram and this diagram are probably the most important right now. This is where we're focusing on the week. And we talked a lot yesterday in the live session about the growth phase, the synthesis phase, the secondary growth phase, and we call that interphase. And then we said there's a division process that's called mitosis, and then there's the actual separation of the two cells called cytokinesis. And so this is a process that may occur over a few hours, and it may occur over a course of like 90 days. Your blood cells live a very long time. There is a great video clip. I think this clip is in today, so we'll take a look. The rest of the slide presentation was mostly just putting everything I mentioned, because in my live session, I kind of hung out in between these two slides. But a nice, refined, well-written recap of what I talked about is right here in the identification of the phases, the role of each phase, and then this will get into mitosis, which is pretty much where I left, which is just knowing the order: prophase, metaphase, anaphase, telophase. I did talk a little bit about metaphase because of the way the chromosomes are arranged, just to get kids to think, how does the arrangement have to be in such a way that when the cells divide, the two cells are identical to the parent cell. And so I just kind of focused a little bit on metaphase. I did show this slide. I just wanted students to understand that this is what DNA looks like, and that the coiling, the idea of this copying and making sure that you have a process that prevents mistakes from happening by making a chromosome, this certainly is a lot easier to make a copy than DNA if it were all just strung out inside the nucleus of the cell. And so they wrap around these little proteins. This is not super duper important information, but it does show you how the chromosomes are made. And then we got onto the actual process, which we did not cover. That would be coming up later. All right, so why don't we focus then on today, because that's what the instructions are. So we're day 3. This is Wednesday. Review notes before beginning the POGL. So this is going to be a POGL activity. It's going to go over the structure of DNA, and then it will go into the end there, sort of the cell cycle. So let's take a look at this structure. What I would do here is I would probably, if I had an opportunity, that Amoeba Sisters video, I think would be very, very helpful to have. If you had it open up on a different tab, you could go through that. There's a lot of information that you could use to help you with the POGL activity. So I don't have this currently in a PDF. Oh, actually, do I? I do. Oh, that's good. So this will be very helpful. This is already up and running. So why don't I just quickly erase all these annotations? All right. So today, when you're working on the POGL, some things that I want to make sure that you're aware of and that you see. I want you to, first of all, I want you to definitely know that there are 4 nucleotides. I'm sorry, nitrogenous bases. I should call them correct terms. They are nitrogenous bases, and there are 4 of them. I'm not going to be asking you to memorize them, but you should be pretty familiar with the ones that do connect. A's always go with T's, and G's always go with C's. But what I want you to see is why that happens. And one important thing, because this kind of connects off of what we had in the facilitated diffusion in the last unit. Do you remember we talked about gated channels in certain chemicals that maybe have the same shape as that sort of gated channel, and unless that chemical sits there and opens up the channel remains closed? Well, it's kind of the same thing here. You'll notice that adenine has this point to it, and thymine has this indentation, almost as if this fits right in that slot. And that is why those two go together. The other, and the same with guanine, you can see it's got the circle here and this here, so you can see how that would go there. So another thing that I want to make sure you understand is that DNA, when it makes a copy, what it basically does is it takes this and it breaks this apart. All of these break apart. And as they break apart, this side of the strand, so this side here, makes an exact copy of this side. And this side on the left, so we'll go up here then, this side on the left makes an exact copy of this side. And to make sure that they do it in such a way that there's not a traffic jam, one side, so let's say this left side, goes in this direction. So it's the new nitrogenous bases being added are going in this direction, and on the right hand side it might go in the other direction. It's kind of like you and I starting at 2 opposite ends of a football field, dropping cones as we go along, but you're going one way and I'm going the other way. And so the only problem is when we meet in the middle. And interestingly enough, if you kind of extrapolate this, all right, so let's see if we can visualize why chromosomes have sister chromatids in this process. Let's just try to visualize this. It's not actually what happens, I don't think, but I think the way I show it makes it more palatable for understanding what sister chromatids are. So let's just for example, okay, let's just say we have a DNA here, all right, and let's just say these bases already broke apart. So I'm just going to put these little lines here. So these are those little runs on the ladder. All right. Now, copy is being made in this direction here, and this copy is being made in this direction here. So now we're going to go ahead, and we're going to go right up to here. And we're going to, okay, so we're making our runs here. Copy. You can see we got a new strand. Oops. A new strand of DNA. So it's being made, new forms right here, right here, right here. And then this one is going in this direction. So it's going, it's going down, down, and it's making the copies as it's going down. It's chemicals coming in here, making copies, all right? And then if it is that we're meeting in the middle, maybe we could just visualize that that's where the centromere is. And maybe then this makes sense as to why this here and this strand here joined at the centromere are called sister chromatids, because they're duplicate copies. Sorry for the chicken scratch here. They're duplicate copies of each other. So now when you look at a chromosome, if you can kind of visualize this process, it kind of makes sense why chromosomes kind of look like they do when they're paired up as sister chromatids. So I just wanted to throw that in there. I thought it was a way to kind of visualize this. So the POGL will continue on with that process. And some of the things that I just mentioned to you, parallel, anti-parallel, are they both going in the same direction, or are they going in opposite directions when DNA forms? Think about what I just did here and how I formed this ladder going this way and this ladder going down in this way. And we're meeting in the middle. Let's see. Hydrogen bonds. So those are the bonds that form between. That's a term we've seen before. Hydrogen bonds came from Unit 3. Here again are our letters, and we call them complementary. So here's our A, T, C, C, A, and G. And so based on those shapes that we just talked about, how would they combine with one another? So what would the other sequence be? The idea of double helix, this is what we're referring to the structure of DNA. Here's a way better refined process. This is what I tend to do. I tend to do a little bit of an instruction piece, but then as I go through, you can see that this is way more refined. But here you have your DNA. So you got this, here's a side, and it's breaking apart, and you can see that the new nucleotides are coming in. And as they come in, they're going to be attaching, and you have this side here, which would normally be attached to this right here, is basically recreating this right there. And so it'll end up with one run over here, or one ladder up here, and then another ladder going in the bottom. And you can kind of get an idea of maybe being joined in the middle by the centromere. Try to visualize that. This term here, semi-conservative replication. That's an important word there. The key is in semi-conserve. Conserve. Not the conservative so much, but conserve. Semi-conserve. Semi-conserve. Think about semi. It doesn't mean whole. Conserve. I'm keeping part of. Keeping part of. Keeping part of. What am I keeping part of in this picture? As I create one strand that goes off this direction and a whole new DNA strand that goes in that direction, duplicates. What am I keeping part of? This diagram, it just really gives you an idea of the kinds of chemical structures that we share among different organisms. And then it segues into that cell cycle. So now you can see why we took yesterday's DNA structure, elaborating a little bit more on it, giving you some more refined practice with it, and then getting you back into that cell cycle. So now you have a really good understanding of what's actually happening in that synthesis stage. That DNA structure, what goes on, the copying, all of that is happening in that synthesis stage. So this whole first part of the POGL is about what goes on right there. That's it. It does go up to the end to question 20. So let's go back. All right. So in addition to the notes, you also have the chapter. There is, I'm pretty sure this is an edulastic that you're going to have to do. Let me take a look. So let's just double check. And that is DNA structure and replication. DNA structure and replication, yes. So I will just double check, make sure I put that link in there for everybody. If you have any questions, please feel free to reach out. Today is Wednesday, so we're available from 9 to 11. It's unstructured time. send me an email. We can hook up for maybe a one-on-one extra help session. I am not available from 9 to 10. I do have a student I'm meeting with, but from 10 to 11, and even after that, if you just let me know, we can arrange a time to meet. Otherwise, feel free to reach out to me via email, and I will have a live session tomorrow, and we will review this activity again in a little bit more detail. So if you have questions, please come with those questions tomorrow. Thank you.